And so then I'll just highlight a couple of things that the Go-To Foundation for Lung Cancer is doing. So hopefully many of you were able to participate in the webinar that Dr. Jeff Oxnard at Dana-Farber Cancer Institute did for us on the study we have called Spacewalk. Raise your hand if you had the opportunity to see it or know about it. So, or raise your hand if you've heard about the Spacewalk study. Okay, great. So that makes my job easier. And so this just reinforces, I kind of wear two hats at Go-To. We have a sister organization called the Adario Lung Cancer Medical Institute. And ALCF, which is part of the Go-To merger, was founded in 2006. Alchemy, that's what we call it, was launched in 2008. And it was out of the recognition that we needed to do more. You know, ALCF was very patient education focused. We needed to do research. And so this is our research arm that does all of our clinical studies. And so Christine is PI of one of our ROS1 projects, developing models for ROS1 lung cancer, and works closely with us. But Spacewalk is led by Dr. Jeff Oxnard. And you heard the reasons why. We know that these drugs only work for so long until resistance occurs. And so we want to understand those mechanisms, what's going on, why does resistance occur, and then use that information to design new drugs that can hopefully continue to help patients. So this is a blood-based study. It's a virtual study, meaning you can participate from the comfort of your own home. We actually make mobile phlebotomy part of this. So we want to bring the study to you. We want to make it as easy as possible for you to participate in this study. And so the idea is that by taking some of your blood, we can look for those resistance mutations that are occurring and use that information to drive forward research on new therapies. So it's a very exciting study. You guys have been phenomenal. I mean, Colin and crew getting the word out. The ALK positives, you cannot underestimate the value of what you guys have done, just talking among yourselves, helping your peers know that this is available. And because of that, you know, the study opened in March. We already have close to 50 patients participating. We've had over 140 calls, and that's because every time you guys share it on social media, you know, our nurse Alicia, gets a call. You know, somebody's interested or asking a question, asking if they can participate. So please, please, please know that that study's out there. It's still, you know, our goal is to accrue 300 patients. We're well on our way, but we need all of you sharing the word, participating when you can, asking the question, asking your doctor. We have a dedicated hotline that you can call. I have flyers out on the registration table. Please come up to me at the breaks. I'm happy to share information with you. But, you know, back to what Christine said, we want to understand those molecular underpinnings. We want to understand why resistance is happening and figure out how we can stay one step ahead of the disease. It really is kind of this evolutionary battle between the cancer and the drug, and the cancer is trying to outsmart the drugs that we throw at it. So it's a great opportunity. Thank you for your support and engagement on it. We want more of you, so continue to share, and if you have any questions, please let us know. The other piece that I was supposed to talk about is how many of you have heard about our lung cancer registry through Go-To? Okay, so a number of you, and that's another easy way. I mean, the bottom line is that research doesn't move forward without patients, and patients are key to this. And so the Lung Cancer Registry is one easy way for you guys to play a fundamental role in advancing research. And you can do it from the comfort of your own home, you know, on your computer. You just go to lungcancerregistry.org, and it's our effort to basically collect data on lung cancer patients. So currently we have over 1,600 patients, a number of ALK positive patients, fortunately, in the registry today. So thank you for your participation there. We need more. And this is where we collect a baseline survey. We just want to know your basic demographics as well as the specifics of your case and your current and prior treatment. But on top of that, then we're working closely with patient groups and others to build additional surveys, so IRB approved surveys. And we've been working closely with Colin and other members of the science and research team to develop an ALK dedicated survey. And so we continue to move forward on that work, looking for what are the specific research questions that will benefit your community the most? And that is a dialogue that we're having with multiple groups, because we really want it to be something that delivers value and moves that needle for you guys. But there, Upal talked about quality of life, and that's something we're building in longitudinal modules, quality of life indicators. The questions around treatment sequencing, that comes up quite often. People want to know, what does it look like if I take this drug first versus that one? And, do I get better duration of response? So there are lots of things that, as a field, we still want to know the answers to, and this is our mechanism to do that. The Lung Cancer Registry is a highly, useful research tool in that regard, and we can ask lots of important questions. So you guys are very much part of that process, and we thank you for your participation. And please, go to lungcancerregistry.org, check it out, ask questions, but we can't move the needle without you guys being involved, and that's an easy way for you to do that. So thank you.
We're going to answer questions at the end.
So just a quick follow-up on that, to piggyback, that you're going to hear a lot about the NCI ALK Master Protocol from Alice when she gets here. By the way, Alice is amazing and certainly the biggest star in ALK positive lung cancer. So I'm going to save that discussion to her, but also say I want everyone to know that for us, when we're researching, we are all friends and we work together, and this is really important because nobody can study this alone. Just like you have a big community and we are part of that community, we share reagents, we share cell lines, we share patient samples. Not one university can do this by themselves. Not one cancer center can do this by themselves. And so that, just really want everyone to know that you may see different physicians up here talking about different research projects, but there are huge groups of people going on behind the scenes and together to really make this, it really does take a village. Research is not one person in one lab studying one protein. It really is a huge community. So I'm going to talk to you a little bit about some ongoing efforts in my lab, and then also in collaboration with Trevor Bavona, who you saw a picture of before, who is a lung cancer researcher at the University of California, San Francisco. So I will say I'm really interested in understanding what makes the wheels turn. Why did that ALK positive tumor cell become an ALK positive tumor cell in the first place? And from my perspective, I think we can put a lot of Band-Aids on things in medicine and science, but until we understand why the disease state, in this case, ALK positive lung cancer, happened in the first place, we're not going to be able to really develop and deliver the most effective therapies. And I will say there's two really big components. There's thinking about the tumor, which is kind of the perspective I come at, is what makes the wheels turn. And there's thinking about the host, which is how does our own individual immune systems play into the biology of the tumor. So from my perspective, in my lab, we only use patient samples and human cell lines, and so we have made cell lines that are ALK positive from numerous ones of my patients, have shared those around the world, have gotten other cell lines from other people, have made cell lines that were sensitive to different ALK inhibitors, resistant to them, so I can take a cell line in the lab from a patient who's never received an ALK inhibitor before and make it become resistant in the lab, and that's a huge wonderful tool for us to be able to ask then, what changed? Why did a tumor cell that was sensitive at one point become resistant? And there's lots of ways we can do that. And that's very effective. And more than that, we can say, okay, I'm not just interested in thinking about alectinib resistance. I want to think about lorlatinib resistance, ceritinib resistance, brigatinib resistance, crizotinib resistance. What are the similarities between resistance to these different ALK inhibitors? What are the differences? And actually tumor cells actually send messages to turn up or down immune systems, and we can detect that by just studying the tumor cells themselves. So I will say, from my perspective, I'm really interested in what makes the wheels turn, and the ways we do that are a couple different ways. First, we do things, and science is amazing. I think everyone said it so far in this panel. Science is so cool, and I love that Upal calls himself a nerd, because just science is amazing, and we have the ability to do things now. Imagine this, if I took a cell line from one of my patients, and I have the ability, that cell line has thousands of genes in it, right? So it's not just ALK. It's not just PD-1 or PD-L1 or pick your favorite protein. There are thousands. And there are ways in the lab where we can actually genetically manipulate those cells. So we can individually, on a one-by-one basis, knock down or turn off single genes at a time, and then ask, if I silence that gene, that one gene out of thousands, does that affect how the ALK positive tumor cell grows? That allows us as researchers to very systematically say, there's 5,000 genes in this tumor cell, and I'm going to knock them down, turn them off one by one by one by one, and ask which genes, when I turn them down, affect how this ALK positive tumor cell grows. That allows us to really get at these questions of what are the wheels that make the tumor turn? And there's ways we do that. You've probably heard CRISPR is one that's a lot in the media right now. CRISPR is a way to actually turn off a gene on a one-by-one basis. There's other ways we can do that too, something called a siRNA, which is basically a way to just turn off a gene one by one. And this is not just for ALK-positive lung cancer. We do this for all sorts of cancers in the laboratory now. And in my lab, we're taking that sort of information, starting with cell lines from patients that are sensitive or resistant to the different ALK inhibitors, knocking down these genes one by one, and then matching that information to what we call high-throughput drug screening, where we can take cancer cells that are either sensitive to the ALK inhibitor or resistant and treat them simultaneously with thousands of drugs, many of which are already FDA approved, and say, okay, what drugs in combination with an ALK inhibitor will make this cell die more? And so if I want to come up with combinations, what are my goals? I want to understand the biology so that I have rational combinations that make sense based on the biology. And I want to facilitate moving that combination into the clinic as quickly as possible. And if we can use drugs that are already FDA approved, from our perspective, as physicians and researchers, that makes it go much faster because the hill is a lot higher to climb when it's a brand new drug. It's not impossible, but if we want to think about rapid translation, using drugs that are already FDA approved makes it much easier for us. So again, very interested in what makes the tumor wheels turn, how can we understand on a gene by gene level, what are the ALK partners, what cooperates with ALK to make ALK tumor cells grow? Can we repurpose some already FDA approved drugs to actually either prolong the response to the ALK inhibitor or overcome resistance? Those are kind of the big themes. And Trevor's part of this too. And hopefully you'll all hear him talk at one point. He's delightful. He has a dog named Mr. B, which just, to me, I just think that's so charming. Really, like, we are all friends and we know each other and our families, and it's just such a fantastic community. Trevor is also developing a lot of what we call patient-derived organoids, where it's a different way to take a patient sample and make it grow in culture. And I will say, from both Trevor and I, what we really believe very passionately is go directly to the source. Study the patient tumor samples themselves. And this is an area where you all can help us a lot because it is a big challenge to actually get tumor samples and to get viable tumor samples that we can bring back to the lab. But Trevor is also very interested in making patient-derived organoids a way to study patient samples and really understanding what makes the wheels turn. And he used some of the same methods I do, but then uses some different ones. We put them all together to really ask these questions of how do we understand the biology? How do we bring rational combinations to the clinic as quickly as we possibly can? I think before I end, there are a lot of nuances to how research goes or how we execute research and things that I'm still learning every day now, having run my own lab for 6 years. I am happy to chat about any of that stuff and tell you sort of lessons learned because the obstacles for us too are very important to think about. How do we get over it? How do we get under it? How do we move it apart? Because we can't advance the science and the medicine unless those obstacles are put aside, and there are some big ones. And we appreciate your help very, very, very much. And I will tell you, federal funding for lung cancer is low. So the most common disease in our country, the highest cause of cancer-related mortality, the lowest funding rate, cancer by cancer. That's not acceptable. And then I will tell you honestly too, a lot of times what we'll get from grant study sections is, this is not common enough to warrant giving you federal money to study this. That's not good enough either. What we learn from studying one cancer type translates to others. And lessons learned are not just for ALK or just for EGFR. They will inform other cancer types, not just lung cancer. They will inform other molecular cohorts. What we study in ALK may help patients with ROS1 lung cancer. So this is really important, and just can't emphasize to you all enough how much we appreciate the partnership in making sure that these obstacles for advancing lung cancer research are completely disabled and we just knock them down together. Thank you.
So as I mentioned earlier, my research focus really is on the cellular and immunomodulating therapies some of which I mentioned earlier, particularly T-cell therapies and NK cells, or again, another subset of promising cells that we're looking to really amplify and re-engineer to improve the immunotherapy response in ALK patients. In terms of an update on the other area of interest would be the combination with radiation. So the Bright Star trial that I mentioned, we started that really accruing in earnest just the first of the year, I think, and it is accruing extraordinarily well, and I have no doubt that is in large part due to the effort that you guys have put in the ALK positive group in terms of just listing it as as one of the clinical trials that are available, and so and we're eagerly awaiting some of the surgical specimens that we've collected and blood specimens that we've collected to look at the early data to see how people have responded to the brigatinib, and then also, in terms of partnering with the ALK positive group, I am just so excited, especially given the big data background that I have, to see how we can leverage, particularly in an ALK-specific way, this lung registry and other efforts, because that is just a treasure trove of information that can help generate new research ideas or to validate research questions. For instance, people who have an exceptional response to crizotinib, alectinib, brigatinib, what may be different about those patients? Or even just to give us a little bit of a better signal about, another open question right now, which is, brigatinib after alectinib. Is that, a viable possibility? So this real-world data that is encompassed in your collective clinical experiences is super valuable. So.
I guess I have the honor of ending the panel, so I'll tell you a little Colin story, and I have lots of Colin stories. So Colin called me up a few weeks ago, and he said, "Oh, I did a survey. I did a survey of our community, and I have this data." And he said, "Will you help me analyze it?" And of course, he knows I'm not going to say no, because data and me, I never say no. I love analysis. So Colin asked a very, very simple question to the ALK positive community. When were you diagnosed? What's your age? And did you have brain metastases at the time of diagnosis, or when did you get brain metastases? Simple 3-4 questions. And of course, if you're male or female. And Colin shared this data with me, and then we analyzed it together. And again, very simple survey. We analyzed the data together, and we saw something very interesting, which is that patients who are diagnosed younger, they tend to present with brain metastases a little bit more than older ALK-positive patients. Now, very interesting finding and a very important finding. Now, the reason I share this story with you is it's extremely important to actually do these types of survey studies, because as a community right now, all the information that we have about the ALK-positive community, in fact, the lung cancer community in general, most of that information comes from clinical trials. And clinical trials are great, but patients who are parts of clinical trials, typically, they do not represent the entire lung cancer community. So the survey that Colin did was actually a very good starting platform to actually go ahead and ask this fundamental research question: does the age of diagnosis impact brain metastases? And that's why I think as a community, we should be doing more and more of these studies so that we can identify these critical research questions that then we're able to study and actually then impact the community.
So we'll give a huge round of applause to the panel here afterwards, but I think we've got time for questions and answers for a little while. And so I don't know if there's a microphone that can be taken to the people asking the questions. And if you wish to direct your question to a particular speaker, please do so, or if it's just a more generic question, we'll pass the microphone to whoever wants to answer.
Hi, my name is Rick Meyer. I have stage 4 lung cancer. My question is basically about spacewalk. I don't know how many people are aware of the really tight time restraints for the actual testing, that you have to have progressed, but prior to starting a new treatment. And I just thought it would be good if you guys clarified that, because I know people who have not been able to do it because by the time they were thinking about doing it or finding out about it, they had already started another treatment, so they were no longer qualified.
Right. Yeah, I mean, that is a reality of the way the study is designed. I don't want that to dissuade anybody from asking about it or looking into it. So, you know, I still encourage people who are ALK positive to, you know, contact Nurse Alicia and just see if you qualify and if you meet those time constraints. And, you know, so it is kind of case by case. I mean, that is a challenge of the study. I mean, I acknowledge that fully, but, you know, I think it's still an important, worthwhile study for the community. So, again, I just encourage everyone to call in, get the information, and see if you qualify or not based on your specific case.
I'm just saying I want people to understand
Right.
that it is time sensitive.
Right.
And, you know, that's something to jump
Right.
on as opposed to...
Absolutely. Yeah. I mean, you know, back to this point of when you know that you're ALK positive, I think it's something that we want it to be on everybody's radar, you know, so that you can be proactive and engage with your physician and say that, you know, it's your desire to participate if you are eligible. So, yes, we want everybody in the ALK community to know about this study and to have that conversation with their treatment team as soon as possible. Yes.
Might be able to talk loud enough. Is the spacewalk just for progression in the body, not the brain? And I have another question.
Yes.
Okay. Why is that? Curious.
Do you want to, I mean, Christine might want to speak to that as well, but I think, you know, it's talking about, you know, blood and whether or not the tumors can shed, and can we detect those mutations in the blood. And so we have greater likelihood of detecting DNA from tumors in the blood, you know, in your body than, you know, from brain mets. And so...
Hearing like this morning that tissue is very, very important. How would it be better to extract the tissue than necessarily DNA, or are we just working on more?
Well, I think, you know, we always say tissue is the issue, right? And that is very precious tissue. And, you know, the first priority is using what tissue is available for your diagnosis and for everything that, you know, to make sure you're on the appropriate treatment regimen. And then, you know, this is kind of what's left over for research studies. And so this is, you know, we are in the age of kind of liquid biopsies and moving that forward. And so it's trying to utilize what is available to us so that we can ask those questions, recognizing that, as you saw on that one slide, I mean, you were talking about such small amounts of tissue in some cases, and tumors are very heterogeneous to begin with. So, you know, tissue can be complex, but this is, you know, Dr. Oxnard is one of the leaders in kind of the liquid biopsy space as well. So, and the company that works with us to do these tests is, you know, at the forefront as well. So that's kind of the reasons why it's blood focused.
So the second question I had, I guess, is to Christine, correct? Very important. You were talking about taking each gene and turning it off one by one, which would require tissue, correct? That would be the most important aspect.
Yeah, so the way we work with tissue is, so I can take a biopsy sample if there's enough, or better yet, pleural fluid. When you're draining pleural fluid, that is the easiest way for us in the lab to make a cell line that we can propagate, meaning keep growing for years. I have cell lines from my patients from 2012 that are still growing in my lab. That is the best case scenario, because it becomes a resource that is available to the community, that we can't get rid of, that just keeps, once a cell line is made, and I welcome any of you, if you want to come and visit a research laboratory, every single person in this room, I would love to host you in my lab. Gina and Amanda have been to my lab, would love, love, love to have you, because I think it is really helpful to see what does research look like in action. And so, yes, tissue is, I will say, always our best source, and I want to comment on that as well, because it's a really important issue. For the lab and turning off the genes, we take cell lines that were derived from patients that can propagate, that can grow indefinitely, forever, and just keep growing and growing and growing, and we use those for our studies. Now, the bigger issue of tissue, and this is a big issue that comes up a lot, and I get a lot of questions about this. I'm sure you all have a lot of questions about this. The biopsies we get, when you go for a core biopsy, they are so small, I can't even show you or tell you something that is a similar size. Maybe like the most analogous size I can represent is, think about a fine tip from a pencil. Like, think about pencils you used when you were in school. And there's these little itty bitty bitty, very narrow, very small tips of the pencil. That's what a core biopsy looks like when it comes out of a patient. And oftentimes, most of that sample is not even enough just to get what we consider standard of care testing, meaning a pathological diagnosis. The pathologist looks at it, meaning the standard of care genetic or molecular testing, never mind research. So I will tell you, the dropout rate for actually being able to, if a patient has a biopsy, in my experience, if a patient has a biopsy and we ask them, can we get extra tissue for research? About 50 to 75% of those times, we cannot get samples for research because there's just not enough tumor material. So tissue is always, always, always the issue, and accessing it any way we can. And liquid biopsies is a huge area of research too, very important, and would love to discuss that more if there's questions.
So if you radiate that spot, breast, and then they radiate it immediately, you're not helping the research.
So I think I will say the patient always comes first. And so what makes the most sense to be the most helpful for the patients that we're treating, that has to be the priority above research. Absolutely. Research is great. I love Gina's research equals life, but the life you're seeing in front of you is the most important thing. And so that has to be the priority. Research is not our first priority. The patients are our first priority. And so we have to do and always want to do what makes the most sense for them. If we get extra, that's great. And I will tell you, we have tissue from patients who've received chemotherapy, radiation therapy, whatever. We keep that in mind as we're designing our studies because it's going to change the context, right? But it's not, anything is valuable in my opinion.
We have time for one more question, and then we are going to have to break for lunch. Corwin, did you?
Thank you. My question has to do with the accumulation of data. In the age of big data, how do we keep from narrowing the data we're collecting down to individual studies and work to collect enough data in a broad enough spectrum of patients that we find things that we would never have thought to look for?
I'm going to say a quick answer, then I'm going to hand it over. So you will hear about the NCI ALK Master Protocol, which will be a study that's open at thousands of sites across the country, and I think this is a really important, great example of collaboration to get big data and make it accessible to across the country. And so stay tuned, because Alice is going to talk about that.
I mean, I think that's really the crux of the issue, because in order to find significant meaningful, statistically meaningful things, you need a bigger data set. And so that's why the work by groups such as GoTo are really critical in trying to bring in real-world data in which we have better access to some of these, some of the data. And then also, as Christine mentioned, things like the ALK master protocol, which are run through cooperative groups, that's where we can really collate and compile that data in a much broader fashion. But you're right, I mean, the sharing of data across studies is an ongoing sort of to-do.
I would just add one more thought. I would just add one more thought there because, we want to reinforce that we guys are friends and the research community is a close network. And so, we're sensitive to patient fatigue. We're not the only survey. I mean, there are multiple efforts, people asking questions, and, we have to think about how do we maximize and strategize and coordinate and collaborate so that we're not asking redundant questions, that we really are, utilizing your time and your resources, to the highest and best use. We don't take your participation for granted. We do keep that always at front of mind is how can we deliver value to the patients, but not also just, wear you down to the point that you don't want to hear from us again. I mean, that doesn't help any of us. So we do, work and think about that as well.
I'll just add one comment, and I think this is, again, maybe the nerd in me which thinks about all of this, but I think sharing data is good, but sharing doesn't mean that the data is usable. I think there are a couple of steps to getting to that point of sharing. Is first, are the different groups collecting data that can be shared? And secondly, is this data in a position that can be mined? And I think we as a community, we become much better at figuring out some of these earlier issues, that we are all asking data that's analyzable, can be curated, and then most importantly, can be used.
Thank you.
So, hands up anyone that thinks research is important? Anyone that wants to say, go research? Research. And surveys are research, and before too long, we're going to be putting out a survey, and everyone thinks research is important. Everyone realizes surveys is research, so please, please, please do the surveys. And I think we can all agree this was absolutely phenomenal. Thanks to the 4 speakers.